ஸ்டார்ட் பண்ணுங்க.
Happy welcome to all. Finally, we are at the end game of our webinar series. To make it special, here we have Dr. S. Prabhu, Associate Professor, Sri Venkateshwara College of Engineering. He has a strong foundation of 19 years of experience. He has published our 19 plus 1 books. His area of interest are biochemistry, molecular biology, genetic engineering, nanobiotechnology, immunology, cancer biology, tissue engineering, medicinal plants, free radical biology, antioxidant therapy, and industrial biotechnology of areas in research. And to add a feather to another one's cap, he has an achievements and awards a list piled up. He was awarded JC's Award for highest marks scored in SSLC in second school high school level, Vellore district, Tamil Nadu. Best Student Award for Proficiency in Studies, Biochemistry from Ismail College, Vaniambadi, Vellore district, Tamil Nadu. 5th rank among affiliated colleges in BSc Biochemistry under University of Madras, Chennai. Best Paper Award in National Conference and awarded prize and certificate in SRM University. The Marcus Who is Who in Medicine and Healthcare Award in 17th edition of US Publication Board. Certificate of Appreciation Award for mentoring the students for the SVCE Student Research Competition held on 2014 at SVCE. Best Research Paper Award in Research Competition held on Intercollege Research Day during the year of 2013. Indian National Academy of Engineering INAE Innovative Student Project Award 2015 for mentoring the project for B.Tech students. Thank you, sir. Thanks for accepting our request and joining with us. Now the session is handed over to you.
Thank you. Thank you for Nagi's introduction. I would like to thank the HOD, Head of the Department of Biotechnology, PSR Engineering College, Dr. Subramaniam sir, and Dr. Rajeswari madam, and Karthi sir, and Venkatesh sir for giving me the opportunity to present this particular students program during the COVID-19 pandemic lockdown situation. So all the student participants, they are very much tired about after attending all these 4 days or 5 days program, and myself is the last one, and I think you are all very eager to listen and observe cell technique, stem cell technique, because many of the participants or the students are young generation. They are showing more interest in stem cell research rather than remaining animal or remaining in silico or in vitro type of studies. So with this introduction, I would like to go for the current topic, modern perspectives in stem cell techniques. So I want to know the differences between the stem cell technique and normal general cellular techniques. So you can I can give you the introduction simply. What is cellular techniques and how it is different from stem cell technique. Okay, so you have to properly know what is the use of stem cell rather than normal cell. You have microbial cell, and you have plant cell, and you have animal cell. And what is the significance of stem cell? Why do you want to study about stem cell? And what is the current strategy using stem cell technique for the treatment of different type of untreated diseases? And what is ethical related to stem cell technique? All this information we can cover in this particular topic. And a brief overview about the current modern perspectives in stem cell technique is, I'm going to define what is stem cell, how it can be classified, what are the sources of stem, from where you can get or produce a stem cell, and what are the subtypes of stem cells, and how do you characterize the stem cell? You have to differentiate normal cells from stem cell, and what are the different type of characteristics of the stem cell, as well as how it can be prepared, even if it is a different type of stem cell. Each stem cells are prepared in a different manner. So how you are going to prepare each different type of stem cell, how it is processed for particular application oriented movement, and what are the different type of application of individual stem cell in the modern era. And even if it has more application, some of them stem cell techniques are also making some disadvantages. Even it has advantages, some disadvantages also there, some ethical concerns are so there. So all this information we are going to see in this particular topic. And the whatever you are seeing in the screen, it's nothing but your already know matured eukaryotic cell. Already any all the even primary students and those who are studying in 5th standard, 6th standard, these students will be knowing what is a matured eukaryotic cell, what is the difference between prokaryotic and eukaryotic cell. When you talk about eukaryotic cell, it is containing different organelles, and each organelles are shown here. And I can show you different type of organelles. It's the powerhouse of the cell mitochondria, and the centriole, which is useful for replication process, so that is mitosis and meiosis. And cytosol is the place where all type of organelles are present. And you need rough endoplasmic and smooth endoplasmic reticulum for protein translation. And for synthesis of protein takes place in ribosome. This is a protein factory. And you have the lysosome for degradation of the antigens. And Golgi complex are responsible for the movement of the signal or processing of transcription processing of the molecules and protein molecule before DNA or RNA is converted into protein. And chromatin and nuclear envelope which are responsible for replication process. So these are all the main applications of the internal organelles of eukaryotic cell. Okay, what is the difference between this eukaryotic cell and stem cell? Since you know all these things are available in eukaryotic cell, stem cell is a initial cell which is not having all these type of organelles, or otherwise it is called as undifferentiated cell. It is not differentiated like this matured eukaryotic cell. So then what is stem cell? It is nothing but it is a master cell. It's a root cell. It is a root cell. It can grow into any type of the more than 200 cell types. It has the ability to divide, it has the ability to differentiate, and it can develop continuously into different type of cells. That is called stem cell. So when you compare with this eukaryotic cell, stem cells, so far it is not divided at its what is called lower level, and it has to be converted into different type of cells by differentiation process. And after differentiation, it is going to be developed into different type of tissues or organs. So the normal stem cell, the root cells are divide, and it has to differentiate, and it has to develop continuously. And simply it can be classified as undifferentiated cell. It is not like this differentiated cell. This is undifferentiated cells, and these undifferentiated cells are converted into differentiated cells, or otherwise called as specialized cell types, by doing stem cell techniques. That is the main difference between normal cells and stem cell. So they are going to be differentiated by its culturing method, that is otherwise called a stem cell culture method, and it is going to be converted into specialized cell type. So this is the main information or definition or differences between the eukaryotic matured cell and the stem cell. So this is an illustration of a stem cell. You have a single cell where you have the nucleus and it can replicate itself. So this single root cell, the root cell is nothing but your stem cell, and it can be divided into two, and this divided cell can be converted into or differentiated into multiple cell. So they are differentiated into multiple sites. Say, for example, it can be converted into the neuronal cell, it can be converted into the muscle cell. So this is the main definition for single cell, which is otherwise called a stem cell, when you compare with the eukaryotic matured differentiated cell. But this is immature or undifferentiated cell. After cell culturing method, it is going to be converted into division and differentiation as well as development. And this is this illustration shows you how this stem cell is converted into, for example, myeloid cell, and the same stem cell can be converted into lymphoid cell, because stem cell can be differentiated to different cell. That's what I told you earlier in the definition itself. So the lymphoid cell, it is nothing but it is responsible for the our immune cell, mainly T cell and B cell. So this even natural killer cells also produced by lymphoid cell. So T cell, B cell, natural killer cells are produced by lymphoid cell. These lymphoid cells are so produced from stem cell. Similarly, stem cell also can be converted into myeloid cell, and this myeloid cell can be converted into RBC cell. It's otherwise called as erythrocyte, and this is obtained from reticulocyte. So reticulocyte formation is done by myeloid cell, which is produced from stem cell. And even the platelets, because our blood cells are containing platelets, and you might have heard in dengue, platelets are reduced. If suppose if you have reduced components of platelet, the dengue attack will be more big, and they are giving some drugs for the improvement or the multiplication of the platelet. So platelets also very important in immunogenicity, and this same stem cell also responsible for the production of monocyte. And this here you could see the lymphocyte, here you could see the monocyte, and monocyte also responsible for production of macrophages as well as dendritic cells. Dendritic cells are otherwise called as antigen presenting cell, and the same stem cell is also responsible for other granulated cells, that is otherwise called as neutrophil, eosinophil, and basophil. So you could easily define now what is stem cell, because of by seeing this differentiation, you could see how this single stem cell converted into type of cells. This is mainly one of the example, hematophytic stem cell. So heme cell, that is otherwise blood cells, are produced from stem cell. It's a very important diagrammatic representation. Okay, how do you classify stem cell? Stem cells are classified generally embryonic stem cell and adult stem cell, and you can modify the corresponding stem cell of adult into induced adult cell, that is otherwise called as pluripotent stem cell. And these three types are generally general classification of stem cell. Even more, this stem cell can also be classified based on sources. Embryonic stem cell can also be produced from the embryo or zygote, and this zygote will have the blastocyst. What is blastocyst? And the blastocyst is nothing but the inner mass of the embryo, that is the inner mass of the cell, and where you have 8 stem cells or cellular accumulation, or otherwise called as total mass of the embryos are present as blastocyst or vessel type of cell, where you have accumulation of this blastocyst. From this you can produce embryonic stem cell. And if suppose if it is not possible to synthesize embryonic stem cell from embryo, you can also synthesize or produce stem cell from adult tissue. Here I have given adult tissue. You have different type of adult tissue that we will see afterwards. While going on the next slides, we can go about this adult tissue, which can be used for synthesis of stem cell. Even amniotic fluid stem cell, or otherwise called as umbilical cord. Umbilical cord are also containing enough amount of or excess amount of or plenty of stem cells. Therefore, they are called as cord blood cell. So amniotic fluid also contain the stem cell. They are called as amniotic fluid cell. So these are all the sources of stem cells. So you can classify based on the general classification and based on the sources. And based on the sources, even the embryonic stem cell, ethically just people are against this production of stem cell from blastocyst, because it is obtained from fetal, and sometimes these fetal sources are available during abortion stage, or otherwise you have to synthesize through in vitro fertilization process. So this is otherwise called as embryonic stem cell. So here the second one, as I told you, is based on source. They can also produced from adult stem cell. So what are the different type of adult stem cell, otherwise tissues, which is having, or which are all the tissues having enough excess amount of the stem cells, are nothing but adipose tissue, bone marrow, and your teeth, which contain the dental bulb, as well as menstrual blood. Female menstrual blood also containing excess amount of the adult stem cell. And the third source is your cord blood cell, embryonic, the what is called fetus embryonic stem cell, and postnatal cell. And this postnatal cell are nothing but umbilical cord you will be having after birth. You will be getting the detachment of the child from baby from mother through umbilical cord, and the umbilical cord will have excess amount of the blood cell, and the umbilical cord also you will be having amniotic fluid, which has more number of stem cell. So umbilical cord contain, as well as the placenta also contain the corresponding stem cell, and amniotic fluid also contain enough amount of the stem cell. So these are all the brief introduction about the sources of stem cell. And what are the characteristics? As I told you already in the introduction itself, the characteristics nothing but when you compare with the eukaryotic cell, they are unspecialized. In eukaryotic, it is developed what? Here, this is unspecialized. This is in starting stage. The cell is in starting stage. That is not specialized into any type of cell. So, stem cells are unspecialized cell. It is not yet converted into the specialized cell till the process of the culture is over. And proliferation capacity of the stem cell is more, or otherwise called as renewal capacity is more, and you can induce this type of stem cell into different type of cells for a long period of time. And the renewal process also varies. That is otherwise called as, if there is any damage to the tissue, the tissue can be renewed by stem cells. Okay, so longer period of time this can be renewed, and proliferation of the stem cells, one of the peculiar characteristics of the stem cell. And this potential, another characteristics of the stem cell is it has many potential. Potential means it has a capability, it has a capacity to differentiate into specialized cell. So potential of the stem cell is maximum to convert it into different type of specialized cell. And plasticity is very important. Some of the stem cell has the potency to convert into different type of cells. Say for example, you can convert a liver cell into pancreatic cell. You can convert a heart cell into lung cell. So the plasticity is different from these different type of characteristics. The plasticity is entirely different characteristics. A particular stem cell which will be producing or expected to produce a particular organ which is stored for the production of a particular tissue can also be converted into a different type of unexpected tissue. That is called plasticity. So I want to say the same explanation. This is otherwise called as unspecialized cell. So you can convert this particular stem cell into specialized cell by making this specialized stem cell into specialized function having cells. So or function doing cells. So such type of fundamental properties are present in stem cell. And it can also divide into 2 or 4 or 8, like that it can be divided. And it can also be renewed, that is for indefinite period, that is otherwise called as long period. And they can also be differentiated under different physiological condition and experimental condition, because our body pH and body temperature is different. So under different physiological and experimental condition, you can convert or differentiate this stem cell into different cells, and therefore they are useful to produce heart cell and heart muscle cell and blood cell or nerve cell, as well as it can also be used to repair the damaged cell in a population of the tissue material. So for tissue engineering, you need three components. One is stem cell, and another one is your extracellular matrix, or otherwise called a scaffold. And the third one is you need some nutrients or signals. And the composition of these three is otherwise called as tissue engineering. That is an entirely different field. But you in tissue engineering, one of the source is stem cell. So the cell is nothing but your ear stem cell. So using the stem cell, you can produce a organ or tissue. As I told you already, explained already in what is plasticity of stem cell. This particular stem cell can give rise to any cell type of cell, or otherwise, say for example, blood cell becoming neuron cell. It's a peculiar character. It's a fantastic characteristics of the stem cell, and astonishing explanation or astonishing expectation of the researchers of knowing this plasticity characteristics of the stem cell, which can convert a particular cell into neuronal cell, which is not relevant to the origin of the cell. Say, for example, blood cell is not relevant to the neuron, but blood cell can be converted into neuron. Liver cells, you cannot expect to synthesize insulin, but liver cell can synthesize insulin. And the hematopoietic stem cell, you cannot convert it into hot muscle cell. You can convert it into the hot muscle cell. So such type of plasticity is one of the major advantage of stem cell. So these are all the characteristics of the stem cell. Okay, then I can go for yeah, many people are telling I have done stem cell line. I have done stem cell culture. Now only I at lab I have done stem cell line, and I have done stem cell culture. I'm coming back to lab. So you might have heard all these words uniformly or differently from your colleagues. So what is the definition for the stem cell culture? It is nothing but, as usual, you are doing culture, microbial culture, or plant tissue culture, or animal tissue culture. This is also a culture, but the difference is the cell is not animal or plant. It is nothing but the stem cell. So this stem cell will come from either, as I told you, zygote, as nothing but embryo, and you can also extract the stem cell from adult tissue. Okay, so what you have to do, as usual, cell you will be growing in a culture dish, and you will be allowing by giving all the nutrients for its replication process. So the stem cell converted into different type of cells by division process or replication process, and this is nothing but culture of stem cell. So similar to other cell cultures, only the difference is here you are replacing the word stem cell. And what's the difference between culture and cell line? The cell line is nothing but, once you get the stem cell, you can collect the corresponding healthy stem cell for division and differentiation, and to develop into different type of stem cell. That is the main difference between stem cell lines and stem cell culture. From after doing this culture, you will be getting number of thousands of millions of culture of the stem cell. From this, you have to pick one particular cell culture of the stem cell, and which one you will collect. You have to collect a healthy culture cell and use that healthy culture cell, or otherwise it is in undifferentiated condition. Now it is undifferentiated but healthy culture. So this healthy culture converted into the corresponding what is called characterized, or otherwise characteristics achieved stem cell. What are the characteristics achieved stem cells? Nothing but divide, develop, and what is otherwise called as differentiate. So if suppose such type of healthy cells are produced or taken from stem cell culture, such type of healthy cell culture can convert to divide, can differentiate, and can also develop, and these stem cells are otherwise called as stem cell lines. So you can choose this stem cell line, and these stem cells alone or subsequently managed what sub-cell culture, substem cell culture. That is, these substem culture are subsequently managed and shared among researcher. That's what you are getting, subculturing method of this stem cell line. So only the difference, you have to take healthy cell culture from this quality, that is quantity of the seed culture of stem cell. That is nothing but stem cell line. And as based on, as I told you already, based on source, based on what is called general classification, you can also classify stem cell into different type of stem cell based on their potential. What do you mean by potential? Potential is nothing but the ability or capability of a person to act like anything. If suppose the actor has a potential to act in different manner, it is nothing but he has the ability to act in a different manner. He can act, he can do fighting, he can do dancing, he can sing music, all these things he can do. That is, he is a capable person. That is what it is called as potential. So the same potential capability is applied for stem cell. So if suppose stem cells are converted into any type of cell, including placenta cell in the zygotic level, that is called totipotent cell. So the cell can be converted into any type of cell. So the potential of the maximum ability of the cell is nothing but totipotential cell. So the ability to differentiate it at all possible cell types, including placenta cell, is nothing but totipotential cell. That is, example is nothing but zygote. And this zygote is formed during fertilization, when sperm and egg fuse together. It is nothing but you are getting fertilized egg. So during the fertilized egg, the zygote formed, it will be having the placenta, and this placenta can also be produced from this stem cell. Therefore, they are called as totipotency cell. Okay, next one is, what is pluripotent cell stem cell? Pluri is nothing but what is pluri? You can convert this pluripotent stem cell into many type of cells. But the problem is you cannot convert this pluripotent stem cell into placental cell. So that you can take any one of the cell type from the zygote, from the blastomia state or blastocyst, and you can convert that particular blastocyst cell into different type of cells. They are called as pluripotent. Pluri means many type of cell. That is called pluripotent. So it can be converted into, differentiated into almost all cell type of. Example include, you have to take this pluripotent cell only from embryonic stem cell. And when you see this embryonic cell, you will be having different layer that I will show you the next subsequent slides. What are the layers? You remember this mesoderm, endoderm, and ectoderm germ layers are present in the embryo. So from this embryo only, these stem cells are differentiated. Such type of potential cells are called pluripotential. Therefore, embryonic stem cell can also be named or discussed, defined as pluripotent stem cell. Okay, what is the next stage? Character, what kind of stem cell? Multipotent stem cell. Multipotent stem cell, here you can use many, here you can use any. This is many, this is a limited one, any type of cell. So multipotent cell, it can be converted to different type of cells. So only the difference is here you cannot use embryonic stem cell, you have to use only the adult stem cell. So that is the main important thing in multipotent cell. So differentiate these two cell. This is otherwise called as non-embryonic stem cell, and this is entirely different from pluripotent stem cell. And here you don't have these layers because this is not derived from what is called embryo. This is derived from only adult stem cell, mature adult stem cell. Therefore, it can be converted to different type of cells, that is called multipotent stem cell. Examples are red and white blood cell or platelets or any type of stem cells. It's nothing but bone marrow stem cell or hematopoietic stem cell. Like that, you can give number of example for multipotent stem cell. And oligopotent cell, oligo means already in biochemistry or in chemistry or wherever, oligo means 2 to 10, 2 to the 2 to 10 number of cells can be produced. Example include you can also utilize the hematophytic stem cell. In hematophytic stem cell, you have the lymphoid cell as well as myeloid cell. As I told you earlier, lymphoid cell can produce T cell, B cell, and natural killer cell. Myeloid can produce RBC cell, macrophages, and dendritic cell, neutrophil, basophil, and eosinophil. So these are all coming under oligopotent. Few cells can be produced by oligopotent stem cell. And when you talk about unipotent stem cell, it's nothing but only one particular cell can be produced from a stem cell. That is called unipotent. A single cell, ability to only produce one cell of their own type, but have the property of self-renewal. But all the characteristics of the stem cells are applicable for all these type of potential stem cell, starting from totipotency to unipotent cell. Only the difference is how much ability of the stem cell it has to be converted into a number of cells. That is otherwise called as the potential of the stem cell. So based on the potential of the stem cell, you can classify these cells into in reverse manner: unipotent, oligopotent, multipotent, pluripotent, and totipotent. Next one. So these are all the description in potential based on the potential of the stem cell. If you take a totipotential cell, it can be converted into a new individual, and the difference is you have to take or expect the particular stem cell only from 1 to 3 days over embryo. So you have to collect the cell only from 1 to 3 days age days over cells. From this particular days developed embryo only can give rise to totipotent cells. So including the placenta, that's what I am telling you, 1 to 3 days. And pluripotent cells, you can produce number of many any type of cell. That's what I told, any 200 type of cell types can be produced from pluripotent cells. And some of the example is blastocyst. This blastocyst is also formed from the zygote, and the inner mass of the layer of the zygote containing the blastocyst. And this is 5 to 14 days aged one. And from this 5 to 14, almost the zygote is developing stage. So from this particular days, you can collect this pluripotent stem cell, that is otherwise called as embryonic stem cell. And multipotent, they can be differentiated but can form a number of other tissues. Here you can say what is called specialized cells. When compared with pluripotent cells, it cannot be converted into any type of cell, but you can convert it into specialized cell. So example is fetal tissue, cord blood, and adult stem cell. Here you have to differentiate by these two cells, totipotent and pluripotent, by having remembering this what is called plasma. So placenta. So the placenta cells are present in can be produced by the totipotent, whereas the placenta cannot be produced by the pluripotent. And the multipotent, it's nothing but not adult stem cell, otherwise called as adult stem cells, are otherwise called as non-embryonic stem cell. And this classification is also shown as diagrammatic representation. Totipotential cell, here the sperm mixes with the egg, and you are getting the zygote, and this zygote containing the multi-layer of the blastosmear. And from this blastocyst, you are taking a particular cell. You have different type of the what is called zygote, and this zygote, sorry, here I should not mention the blastome here. This is totipotential cell. This is nothing but your zygote which is developed into morula stage. And this morula stage has the capacity to convert into total type of cell, whole cell, that is nothing but totipotent. So here pluripotent cell, many cell, here you have the corresponding cellular mass, that is always called as blastocyst. And these blastomeres or otherwise called as inner cell mass. And this, you have the outer layer, placental layer. You cannot take this layer, and therefore you can take this inner cell mass and cell culture petri dish along with the media nutrients. You can convert this embryonic stem cell into different type of cell. So here what you have to give, you have to give signals and some factors, nutrient factors, for the development of this particular embryonic stem cell into different cell. And what is multipotent cell? This is purely derived from adult stem cell or non-embryonic stem cell, and therefore you can take any type of tissue or non-adult stem cell. From this you can convert the same, similar to this, you can give the nutrients and supply of the multiple media, multiple nutrients and media, and it can be differentiated into specialized cell. Therefore it is called as multipotent cells. Sorry, I went to final stage immediately, knowing I pressed the page down button. And 1 second. Okay, this is a differentiation in potency. And next one is your illustration I showed you. And this is also illustration showing potential stem cell. Here you have the single embryo totipotential cells, and here you have 5 to 7 day embryo cell. That is what I told you, pluripotent stem cell. So here you have all type of the blastocyst inner cell mass, and this particular embryonic stem cell you can take for production of many type of cells. And here you can see multipotent cells, multipotent stem cell. Say for example, you can take cord blood cell, umbilical cord blood cell, or amniotic fluid cells, or placental stem cell. And from this you can produce multipotent cells during the birth of the baby. That's what it is shown, the infant. And if you take the stem cell from adult, either male or female, you can take adult stem cell from this particular multipotent. Why it is given adult stem cell, but it indicates infant? But this adult stem cell is only related to the adults, either male or female. Infant cell, it also coming under multipotent, it's shown here, but it is not adult stem, but it comes under multipotent stem cell. So the diagram explaining the information about the classification of the stem cell based on their potential. And these embryonic cells are otherwise called as pluripotent stem cell, like totipotent cell, stem cell in zygote. Embryonic stem cells otherwise called as pluripotent stem cell. So you can produce or synthesize or isolate this pluripotent stem cell from embryonic tissue only. So fetal tissue is the responsible source for the production of pluripotent stem cell, and you can grow it in a culture, but you need some specialized method to prevent them from differentiation. Otherwise, if you not observing the corresponding progress of the pluripotent stem cell, it will grow in different type of cells. So you'll be getting the chances of contamination or chances of differentiation of the corresponding pluripotent stem cell into different type of cells if you don't have any observation or proper observation of their what is called growth, so that you can observe it properly, and you have to add some nutrients while it grows into different type of cells. So that is under your hand, and the contamination should be also be avoided. Otherwise, stem cell research is very risky one. And this pluripotent stem cell, that is otherwise embryonic stem cell, again subdivided into two: embryonic germ cells and embryonic stem cell. So they are produced from gels, gametes after the production of the egg. So that is also your sperm cell also containing the stem cell. Even saliva, urine contains stem cell, but the source of the stem cell in urine and saliva and the sperm cells are very less comparing to bone marrow or otherwise zygote or otherwise blood cell. The source of the stem cells very limited in these sources, as I told earlier. And the another thing is it can also be divided into typed into embryonic stem cell. It can be isolated from inner cell mass. That's what I told, blastocyst. So this blastocyst is nothing but the 8 to 14 week stage of the zygote development. Therefore, the people, some religious people are offending, or otherwise they are against such type of uptake of the stem cell from this embryonic level. So it is unethical, they are telling. So the stage of embryonic development is on progress during the 4 to 14 the day. Therefore, you can take the inner cell mass of the embryonic stem cell, from where you can produce different type of any type of cells, that is called pluripotent cells. So for human, even if suppose if they are not accepting, some religious people or some any other problem of parents, they are arguing for not killing the inner cell mass blastocyst stage of the corresponding what is called embryo, or otherwise called as blastocyst. You can also take another method, alternative method you can adopt. The alternative method is nothing but in vitro fertilization process. What you are doing in vitro fertilization, instead of having the in vivo fertilization process, which is nothing but union of sperm and egg to produce a zygote, the same union of egg and sperm is done in outside. That is called in vitro fertilization, IVF procedure. So during this IVF procedure also, you can collect, you will be getting the blastocyst stage, and you will be getting the inner cell mass, from where you can get this pluripotent stem cell. Okay, next we'll go for, as I told you earlier, to remember how these embryonic stem cells are responsible, or source of cells for the development of the various organs. And as I informed, noticed you, or mentioned you, to remember the layers in the earlier illustration. You remember these layers, ectoderm and endoderm and mesoderm. I showed you a previous picture. Ectoderm, it's nothing but, ecto means outer layer of the embryo. Here I have shown previously. It takes time to rotate to you to show you the corresponding here. Okay, so where is I have shown somewhere. Okay, somewhere I showed you layer. Okay, we'll go for this layer: endoderm, ectoderm, and mesoderm. Ectoderm is nothing but your outer layer, surface layer of the embryo. And this ectoderm is only used if you collect the stem cell from ectoderm. It is responsible for the outer portion of the skin, our body. Our body outer portion is nothing but skin and nerves and some muscle cell and air tissues. Tissues are nothing but produced from ectoderm, outer derm cells. So that is otherwise called as epidermis. And endoderm is nothing but innermost layer of the embryo, which is responsible for the production of the different type of organ, associated organ. You know already, we have different type of organ: lungs, heart, spleen, pancreas, and brain, and kidney, All these cells are inner cells, stomach cell, all these are inner cell. These cells are produced by endoderm. Even in vivo fertilization, if you see in vivo fertilization, you will be getting this type of layer during the stage of the 14 to 15. So each layer are responsible for the production of our different type of organ. So the same thing I am telling here, ectoderm and endoderm. Zoology people, they will be doing well how these zygotes are converted into particular what is called embryo. Biological people are very much familiar with the production of organ or production of human. Okay, then the medical people are also very much know about this. The life science people, everyone, the interdisciplinary of life sciences people are very much knowing about these layers. And mesoderm becomes sandwiched between the ectoderm and endoderm, and this generates a blood, heart, kidney, gonads, bones, and connective tissues. This is otherwise called as mesoderm. So in between ectoderm and endoderm, you have the mesoderm. If you take stem cell from mesoderm, it will produce blood and gonads and bones and connective tissues. Even heart, kidney also produced by endoderm. Mesoderm also responsible. But the difference is depending upon the signal, which enhances are the factor which are enhancing or influencing the layer. That particular layer will be converted into different type of organ. So that depends on your physiological changes present in our body, individual body. And this one is, since we are talking about embryonic stem cell, I'm concentrating on embryonic stem cell and pluripotent stem cell, so that the first stage of the type of the cell, here you have the egg and you have the division of the egg. After fertilization, you are getting this zygote. The sperm induces the egg and you will be getting zygote. So two stage is there, and it becomes a 4-cell stage, and this become morula. And from this morula only you will be getting what is called totipotent cells, otherwise called as including the placental cell, you will be getting totipotent cell, where if you see there's a blastocyst, it's a vessel where you have the inner cell mass, and this inner cell mass otherwise called as a blastocyst, and this inner cell can be used for production of embryonic stem cell or pluripotent stem. So this morula and blastocyst are different. This is totipotential and this is embryonic stem cell, otherwise called as pluripotent stem cell. And similarly, I told you the ectoderm outer layer responsible for your central nervous system production and neural crest and outer surface production, skin and neuron of brain and pigment of the skin. Everything is responsible by ectoderm outer layer, and mesoderm middle layer responsible for dorsal part, notochord, as well as paraxial or skeletal muscle cell, and some of the kidney cells and RBC cell lateral part, and facial muscle of the cell also organ also produced by this mesoderm cell. And in the zygote stage I am telling, and endoderm internal layer which are responsible for the production of tissue or organ, digestive organ, pharynx in your respiratory cell, and respiratory tube where you have the lung cell or alveolar cell, and this particular digestive as well as pancreatic cells also produced, thyroid cells are produced, and the alveolar cells are produced from endoderm. And if you take this totipotential cell, the germ cells, both female and male sperm and female egg are produced together to produce what is called morula, and morula is responsible for totipotential. So this is the information about the pluripotent stem cell. If when you do this fertilization of this egg in vivo, when you do in vitro is different, and whatever the fertilization takes place inside of our body. Once the married couple does intercourse and doing after their menopause period, if you see the testing for the corresponding generation of the egg in the female uterus, you can see easily the 14 to 8 to 14 to 21 days embryo, and this embryo is nothing but produce a fertilized egg, so that you will be getting the totipotential cell including the placental cell. This is nothing but your 8-cell embryo. And when you observe this particular 8-cell embryo, when it comes to 21 days stage, 14 to 21 stage, this is otherwise called as blastocyst. So that this is the placental cell from where this blastocyst inner vesicle, where you have accumulation of the blastocyst are there, and this is mainly responsible for the production of the embryonic stem cell, otherwise called as pluripotent cell. That is the main reason why the people, some couples or parents or some religious people are against this type of generation of stem cell, because already the embryo is developed and it is in the condition of blastocyst, 21 days stage. So they are against such type of monolithy. Therefore, people are against such type of development of the stem cell from the blastocyst. Even though for a research purpose, some researchers are doing this embryonic cell, but the source of the blastocyst is very difficult to obtain unless otherwise the parents or the couples are accepting to take their blastocyst, or otherwise you can get it from aborted zygote. So aborted zygote will have rich amount of pluripotent cell. From that aborted blastocyst cell, you can produce a pluripotent stem cell. And instead of wasting the aborted cell, you can use that aborted cell for stem cell, that is pluripotent stem cell production. So they are undifferentiated cell. When it is going to be induced into different cell by differentiation process, it is developed into neural cell, it is developed into cardiac muscle cell, it is developed into blood cell. So here the differentiation is done by the factors and development is done, and till then it is in undifferentiated cell. So all the characteristics of the stem cells are achieved here and are shown here, as I said earlier. And how do you cultivate? Say, for example, as I told you, in the case of fertilized one, that is in vivo fertilized egg, unless couples or parents are accepting, you can go for alternative option, aborted cell, or otherwise you can go for in vitro fertilization egg. So in vitro fertilization, in vitro, you are doing the fusion of the sperm from male and fusion of the egg from the ovum of the corresponding female, and you will be getting the blastocyst formation, zygote, and zygote you have the blastocyst. And this blastocyst is taken into the petri dish cell culturing, and you will be getting the blood cells, hematopoietic stem cell, as well as blood cells of RBC or WBC or lymphocytes or any type of dendritic cell or macrophages. And you can also produce nerve cells, or otherwise you can also produce the muscles or cardiomyocytes. So here you can say that this one is in vitro, second one is blastocyst stage, and inner cell mass nothing but blastocyst, and cultured undifferentiated cell, fourth one, and fifth one is specialized cell. One is a blood cell and another is neuronal cell and cardiac muscle cell. So this is a stem cell cultivation. And how do you harvest this embryonic stem? Everything is available, the source is available. How do you harvest? How you are going to harvest this embryonic stem cell in lab? So you should develop similar to microbial cell culture, similar to plant tissue culture, similar to animal cell culture. How will you harvest this stem cell from embryonic source? You are available, you have in your hand embryonic stem cell. How will you harvest this? You have to harvest by using this cell culture method. As I told you already, there are two differences between the cell culture and cell line. Only you have to take healthy stem cell from the stem cell culture. From healthy stem culture, you have to take the corresponding stem cell line, subculture stem cell line, and this particular stem cell only you can use for different type of development of the tissues or organ. So you have the blastocyst initial stage, where you have the epidermis, that is outer layer, and it also includes the placenta. And the blastocell, it is nothing but a hollow cavity where you have the 8 number of blastocyst molecule. And this inner cell mass, it contains approximately the 30 cells at the one end of the blastocell cavity, vehicle type of vessel type of the containment. And in the particular vessel, you have at least approximately 30 cells. At the one end of the vessel, you can take called culture cell. Okay, so you will be getting three different type of germ layer: endoderm, mesoderm, and ectoderm, as I told you already. So you have to harvest anything. If you want the endoderm, what type of organ you would like to take or grow, you can take this endoderm. If you want to take intermediate organ, you can choose mesoderm. If you want to develop outer layer, that is otherwise called a skin or bones or nerve tissue or type of skin epithelium or skin type, what is called as the pigment type of cell, you can choose ectoderm. That depends on the corresponding researchers. Those who like to choose the development of the any type of tissue or organ based on their choice, you can choose any type of the layer from embryonic tissue. Okay, in vitro fertilization clinic, they are taking the blastocyst, and that is otherwise called as inner cell mon. That is otherwise is called the blueprint of the cell. Okay, so blastocyst is a hollow ball of cell with a small clump of stem cell inside. That is nothing but 30 number of cells, as I told you earlier. Like our replication process, you need some primer for DNA replication. RNA primer is needed for DNA replication. Similarly, here the primer is nothing but your stem cell is nothing but your blastocyst. That is otherwise called blueprint, and use the blueprint from the inner cell mass of the embryonic blastocyst. And you please isolate and transfer the inner cell mass into plastic culture. You might be seeing many majority of the research people, those who are in doing stem cell technique, they are very much aware of these cell culture technique, stem cell culture technique, and these things are very easy for them while I am expressing. But some people, because participants are mixed people, careful because the growth of the culture, already the cell culture is very difficult. So when you compare with the other cell culture, stem cell culture is very, very difficult. If you have any problem in contamination, the division and production of the stem cell into different organ is very, very risky one. So cell divide and spread over the dish if you provide all culture medium, and inner surface of culture dish is coated with the mouse embryonic skin cell. Here you have to mainly think about the mouse embryonic skin cell. Why do you add? What is the purpose of adding mouse embryonic skin cell into the particular embryonic stem cell? Because this embryonic mouse embryonic, this is other species, okay, other species cell you are adding with the culture dish, where you have your inner cell mass blastocyst along with your culture medium. You are adding the embryonic skin cell of the mouse. Why you need this one? They have to treat with a feed, because they are feeders. This particular mouse embryonic skin cells are giving some nutrients. They are feeding the blastomere of the human cells, so they are called as feeder cells. So feeder cell provide embryonic stems with a sticky surface for attachment, because this produces many nutrients. So you need some, as I told you, what is called in tissue engineering, you need type of support that is earlier I said it is otherwise called as a supporting material. That supporting material, collagen or any type of biomaterial you need for the tissue engineering process. Otherwise, it is called as I forgot what is the correct term. I told material. I forgot the earlier I said. So such type of supporting material are provided by feeder cells. Okay. So these feeder cells provide corresponding. 1 minute. Wait. My secretaries are calling. 1 minute. Hello, sir. Prabhu speaking. Sorry to talk. Sir, regarding that, this one, sir, you sent me the information. No, I already sent you the email also related to that, what is called DBT letter of intent. So which particular theme we have to choose? I think we can choose the dendritic type of. So I send you the message. Mm-hmm. Yeah, 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 yeah. Mm-hmm. Mm. So, mm-hmm. I, yeah, yeah, I saw that. But biologist and some other relevant people there. So we can find out, sir. We can find out now, sir. Yeah. Mm-hmm. Mm-hmm. They are also telling this particular, if suppose if we get sanction, many institute also can utilize our this one, what is called virtual event process, virtual event regional level center. Mm, yeah, I saw that. But whether they mention medical people, I didn't say. I saw that some bioinformatics people and biologists. Okay. Mm, mm. Sir, 1 minute, sir. Participant, I'm talking with my secretary, sir. So please be wait for a few minutes. I'll come. Please wait. Sir. No, sir, no problem. I'm attending the FDP. I'm giving FDP for the students. Faculty. No problem. One. Oh, no, because I tried my best, but that time I thought you went for sleeping, so that it's afternoon time. So I didn't want to disturb you, sir. Hmm. Okay, sir. Okay, sir. I will try my best. Hmm. Okay, sir. Okay. Thank you so much, sir. Thanks a lot. Hello, sorry, participants, because my secretary was on the line and he was discussing about some DBT proposal. So he was asking me to know about the status of the DBT proposal. So that time I cannot avoid by not giving the answer to him. So he was discussing about that. So sorry, I'm extremely sorry to the participant, and we'll continue this one. All are in the line. Can I check? Everybody is on the line. Yeah?
Yes, sir. Yes, sir.
How many?
Sir, you
How many
continue, sir. No problem.
No, no.
No
Yeah, how
problem. All
many
are there.
No, okay.
Okay,
And
no problem, sir. You continue.
okay. Thank you so much. I'll continue. And where I left this, as I told you already, you have to take the original cell mass and you have to transfer into the culture dish, and you will be having all the culture medium, but you have to expand the division of the cells into different type over the plastic dish, and along with that you have to give the what is called mouse embryonic skin cell. Why do you want to provide mouse embryonic skin cell? Because Yeah, now I've extracellular matrix. We are telling in tissue engineering, extracellular matrix. No, such type of extracellular matrix are provided by these mouse embryonic cells, and this mouse embryonic cell also produces nutrients like surface area, and but nowadays providing mouse embryonic stem cell, mouse embryonic cell to be added with the corresponding your what is called embryonic stem. Embryonic mouse cell, feeder cell will cause some infection with the embryonic stem cell. In order to avoid that, recently researchers are not utilizing this mouse feeder cell in order to avoid the infection by viruses or other mouse molecules. Okay, so this is recent information. I got it, and after the embryonic stem cells are cultured, you have to plate into different plate culture dishes, and that is otherwise called as stem cell culture lines. This stem cell culture line will be having that matured or otherwise called as the healthy cell will be produced. So different culture plates from the embryonic stem cell where you did this cell culture technique for specifically for embryonic stem cell. After several days, after several 21 days of the allowing of the blastocyst, or otherwise called as inner cell mass, is converted into or allowed for proliferation, it begins to crowd in the culture dish, the plastic dish. Okay, so what you have to do, simply you have to remove the corresponding culture from the culture dish. It should not be damaged at any cost on the culture of the stem cell, and whatever you have plated layer present on the culture dish, it should be gently removed and plated into several fresh culture dishes. Okay, so this process of plating should be done several times for many months, and that's why I told you the culturing of stem cell is very very difficult, as well as it's very very risky one to get production of the tissue or organs. So this is repeated for several times. Many men you have to wait until then. That particular cell should not be contaminated. So subculturing you have to wait for many months, and this should be repeated. And each subculturing is nothing but people might have saying this word passage. So now only I did the passage, not massage, passage. So passage, then now only I did passage, sir. I came out, sir. So passage is nothing but what? Subculturing of the corresponding stem cell. So usually you are using this word passage for subculturing of the each cycle of the mother cell of the stem cell. Okay, and after 6 months, what you have to do? You have to take the inner cell mass of the blastomia, and in that you'll be having the embryonic stem cell, millions of stem cells, because this is a cell culture technique. Okay, millions of stem cell, and this millions of stem cells are otherwise called as pluripotent stem cell, and they appear genetically normal because you have not done any changes in the genetic molecular level. You have not done any changes in the pluripotent stem cell, so there is no changes in the genetic nature. Therefore they are genetically normal, and therefore they are called as embryonic stem cell line. Okay, so this is otherwise called as healthy stem cell line. From where you got it? Stem cell culture. So stem cell line is different, stem cell culture is different. This pluripotent stem cell line usually normal in genetically it is normal. There is no changes anything in that produced pluripotent stem cell. So this is the illustration showing the pluripotent stem cell. You have the sperm and the egg fertilized and the totipotent stem cell, and you have the blastocyst containing pluripotent stem cell, and you have to isolate the pluripotent stem cell from inner cell mass, and you have to culture it. Whatever I discussed now, this is nothing but your culturing of the pluripotent stem cell. And This blastocyst pluripotent stem cells can also be used for the, as I told, any type of cell. No, this is nothing but hematopoietic stem cell and neural tissue specific cell and mesenchymal stem cell. And you can also produce this for connective tissue or bone or cartilage or nerve system or blood cells. Okay, so these are all the possible kind of proliferation or differentiation or division or development of different type of tissue from pluripotent stem cell after culturing of the blastocyst, that is embryonic stem cell, into the petri dish. And in vitro process, as I told you already, the embryos you collected and it has to be freezed, or the frozen cells are routinely destroyed when couples finish their treatment. Now, tens of thousands of frozen embryos are routinely destroyed. So you either you depend on parents or for getting or for receiving the blastocyst, or you have to do the what is called abandoned for docking studies, which molecule will bind to which particular receptor, and in vitro studies, such type of cell culture technique they are doing. And finally they are going for in vivo study. So instead of going directly to in vivo animal cell culture study, sorry, animal in vivo study, you can carry out tissue culture study. Then if you get success, you can implant these type of cells into the in vivo system, or otherwise called as animal system. So coming back to the in vitro fertilization process, here you have the unused frozen embryo which has been thrown away after treatment for in vitro fertilization process. It is very waste. Instead of wasting that, why can't you choose this wasted tissue for pluripotent stem cell production? So you'll be getting pluripotent stem cell production, and this pluripotent cell can be differentiated and develop into pancreatic islet cells and hematopoietic stem cell, and you can also convert into cardiomyocyte art cells, and it can also be converted into neuronal cell, and it can also be converted into hepatocytes or liver cells. So these are different, many types. As I told you, any type of cells can be produced from this embryonic stem cell. So you can implant these cells wherever you want to implant into the patient. So some people will be requiring the art cell. So such type of art cell which has been produced by in vitro fertilization process from embryonic stem cell can be implanted in this patient, art patient. And those patients affected the liver transplantation you can do by this stem cell, and pancreatic transplantation you can do by this pancreatic. So whichever organ has been damaged, that is otherwise called as degenerative diseases, this degenerative organs can be restored back to by using this stem cell that is embryonic stem cell. And this is the one photograph I have taken from literature. This is not my study, and I have sources from literature. And I have, for your purpose, I have taken this and I have pasted here, and you can easily see, because here in webinar you can take the people to the fluorescent laboratory where you can't ask them to view this type of cells under the fluorescent microscope. Instead, in webinar, you can show the slides of the stem cell, which could be looking like fluorescent type of cell whenever it is produced through embryonic stem cell if you use fluorescent microscope. So you can find out, if suppose if you want to see the embryonic stem cell in fluorescent nature, what you have to do, you have to label this fluorescent stem cell with fluorescent dye or fluorescent antibodies. So antibodies binds with this stem cell, and you will be getting the corresponding stem cell which looks like smaller blue dots in the green area labeled the nuclei of the embryonic cell. So here you have the blue dots. So okay, these blue dots is nothing but your embryonic stem cell. So you'll be looking your embryonic stem cell if you have cultured through this in vitro fertilization or other than in vitro embryonic stem cell culture method, you could see the corresponding stem cell under this fluorescent microscope like this. So it will be looking like this if suppose if you attach this stem cell with antibodies, fluorescent antibodies attached to the fluorescent molecules. Okay, so now this is how the scientist can track which cells are stem cell and which are not stem cell. If some stem cells are not like this, you cannot differentiate these stems. You can differentiate easily the normal cells from the stem cell. So here you can see easily this embryonic stem cell like to grow little colonies spaced out over dish instead of in one big layer. Okay, so these are of cultures of the stem cell, and it's a dish of the corresponding one big layer which is fluorescent. And this is a good illustration or image photo shooted by the scientists in their lab, and they have pictorially represented in a textbook and journal also. So I have taken this and I showed to you how you should know how do you differentiate this stem cell from other cell molecules. And so far we have discussed about the phase 1 type of adult what is called embryonic stem cell. Now we are going for second type of cell, that is otherwise called as non-embryonic adult stem cells. So a non-embryonic adult stem cell can also be called as multipotent, whereas embryonic stem cell are called as pluripotent stem cell. So here multipotent, multi means it can be converted into specialized tissue, whereas embryonic cells can be converted into many type of tissues. And here multipotent stem cells otherwise called as, it is not taken from embryo, therefore they are called as non-embryonic, only they are taken from adult stem cell. And you can differentiate this particular adult stem cell into different type of limited number of type. That's what it is not mentioned any type. It is called as specialized type of cells. So that, for example, the bone marrow contains multipotent stem cell. If you take the bone marrow contains multipotent stem cell, and it can give rise to the all cells of the type of organs or tissues, and the special type of cells can be produced. If you want to produce a blood cell, you can give rise the blood cells from the bone marrow, but not other type of cells. Whereas in the case of what is called umbilical cord, you can differentiate into any type of cells. So here specific type of cells can be that is otherwise called as plasticity. So plasticity is applicable for adult stem cell. So very important plasticity. Here the plasticity is very limited for adult stem cell because it can be converted into a particular specialized cell. That's what it's called, adult stem cell. And multipotent adult stem cell. They are present in adult animals. Example, it can be converted into any type of organ, that is brain, liver, and they can replace dead or damaged tissues. And this particular adult stem cell can also undergo some mutation. That is also very a disadvantage in the case of the adult stem. When these adult stem cells are accumulated together, there are possibility of mutation takes place in the gene of the particular adult stem cell. And while you are getting the clone of the cells, some of the adult stems will become cancerous cells. So that in order to avoid that clone of cancerous cell produced from adult stem cell, you have to avoid the accumulation of the adult stem cell or sticking of these adult stem cell together. So you should be very vigilant about this accumulation of the adult stems together in order to prevent the mutation. in the case of the adult stem cell. So here I told you the multipotent stem cell because we are talking about the adult stem cell, and this multipotent adult progenitor cell can be converted into hematophytic stem cell, as I told you, different type of blood cell, hematophytic stem cell, or otherwise it can also be converted into mesenchymal stem cell. Okay, so if you are getting confused, please go through again what are the different types of classification of stem cell. But general classification is, I can say, based on the source, you can say embryonic and non-embryonic and umbilical cord or induced pluripotent stems. It's not source, but we are inducing. But when you compare with potential, you have different type of potential. So from this multi-potential type only, you are getting the mesenchymal stem cell, and you will be getting hematophytic stem cells. And this endothelial progenitor cells also produced from multipotent adult. So endothelial progenitor hematophytic stem cell and mesenchymal stem cell can be produced from multipotent adult progenitor. That is the main advantage of adult stem cell for the production of these type of cells. This hematophytic stem cell can be converted into used to synthesize blood or develop blood cell and develop endothelial cell, and which one this endothelial progenitor cell can be used to develop endothelial cell, and mesenchymal stem cell can be used to produce neurons and chondrocytes for bones and muscle cells, the osteocytes, bones, and adipocytes for what is called liver or adipose tissue. And these type of mesenchymal stem cell can be used for production of these different type of cells or tissues or organ. You can multipotent stem cell can be used for synthesis of neuronal cell to produce brain, and they are used for cardiac muscle in the replacement of the heart injury or myocardial infarction, and it can also be used for blood cell which is produced from the bone marrow. Bone marrow is nothing but it's very important part of our body which can synthesize immune cells, or otherwise T cells or B cells or immune cells. And T cells and B cells are natural killer cells, lymphocytes and B lymphocytes cells and other neutrophil cells response. So initially the SCID, so there is severe combined immunodeficiency syndrome people, they have been treated with bone transplantation methodology only. So afterwards only they came to conclusion a rich source of these what is called stem cell, which are responsible for synthesis of these different type of blood cell, T cell, B cell, macrophages, dendritic cells, as well as neutrophil, basophil, and what is otherwise called as eosinophils and red blood cells. All these are produced. So bone marrow is a rich source of the immune cells. It's very important. If you lose, if there is any damage in bone marrow, your immunity will be reduced. Even for fighting the coronavirus, we need immunity more. So this type of immunity is produced from bone marrow only. So the bone marrow are very important for a generation of immunity in your body. And this otherwise called as cord cord blood stem cell and hematophytic stem cell, and these are rich sources of hematophytic stem cell and mesenchymal stem cell. Here I told you, here mesenchymal stem cell, MSC cell. This MSC cells can also be produced from multipotent adult progenitor cell, also produced from umbilical cord as well as bone marrow. Okay, so this is nothing but your umbilical cord, and where you have the cord blood. So from this you can produce hematophytic stem cell, and this one what is called cord tissue, where you can produce mesenchymal stem cell. So this is the main difference between the generation or development or differentiation of the corresponding stem cell into different organ. So cord blood can be used for hematophytic stem cell, and tissue can be used for 2-in-1. 1 in 2, and cord tissue, mesenchymal stem cell can be produced. So it can also be called as somatic stem cell, and these somatic stem cells tissue including brain, bone marrow, peripheral blood, mainly PBMC, amount of your monocytes. And monocytes are present more in PBMC, from where you can derive macrophages as well as what is called dendritic cells. And blood vessels also you can get your stem cell, skeletal muscle also you can get, skin also you can, teeth bulb also you can get, heart muscle, gut, liver, ovarian epithelium, and testes. Everywhere you have somatic stem cells. And once removed from the body, their capacity to divide is limited. So that is very important. And if you want to produce, that is very important. And if you want to produce large quantities of, only the source is bone marrow and peripheral blood cell, as well as your what is called umbilical cord. When you compare with the brain, blood vessels, skeletal muscle, skin, teeth, heart, well, all these are, even teeth also will produce enough number of somatic stem cell, but whereas these are all testes and liver, these are all will give you limited number of stem cell. Even that limited number of stem cell should be maintained or cultured properly. That is the main disadvantage if you take other parts of the somatic stem cell comparing to bone marrow and peripheral blood as well as cord blood cells. In the case of adult stem cell, you won't be getting large quantity of stem cell. So here bone marrow is there, develop a blood cell. You can develop brain, you can develop liver. That's what I told you, specialized cell. Whereas in embryonic cell, you can produce a specialized cell. So it can be converted into any type of cell, whereas bone marrow, it can be divided into specialized cell. Depending upon the agent you are utilizing, the bone marrow is developed into different type of cell. Neuron, and can be converted to skin, can be converted to muscle, and this is a rich amount of adult stem cell present in bone marrow. And here you can see this a part of the where from where italic part of the bone marrow stem cell, they will be saying Hittite part of the stone bone marrow stem cells. Here it is lining. This white lining alone, it is projected in large. In maximum projection is shown. So this part of the white Hittite part is bone containing the bone matrix. This is otherwise called as bone matrix. This white fluid, otherwise called as bone matrix. This is extrapolated here, and this white layer containing what is nothing but your nucleus part, osteoclast can be produced from this cell, and blood vessel can also be produced from this cell. And this green color is nothing but your hematophytic supportive stroma, and where adjacent you have this red color. So this color is nothing but hematophytic stem cell, whereas this blood green color is nothing but supportive stroma, and this is nothing but your lining of bone, osteoblast, cartilage. Here the white layer is projected here, this is projected here, osteoblast. So stromal stem cell is projected here, and the pre-osteoblast cells are present, and from this lining of cell are obtained from bone cartilage, and this particular cell are responsible for the production of these type of what is called natural killer, that is lymphoid progenitor cell and myeloid progenitor cell, because they are multipotent cell. So this hematopoietic stem cell also type of multipotent cell like bone marrow, and this is not only in the umbilical cord blood, it is also present in bone marrow. And from this bone marrow hematopoietic stem cell, you can produce the multipotent stem cell of the what is called blood and lymphoid progenitor. From this lymphoid progenitor, you will be getting NK cell, natural killer cell. You can produce T lymphocyte, you can produce B lymphocyte. Whereas myeloid progenitor cell, from multipotent stem myeloid progenitor, you can synthesize RBC cell, you can synthesize platelets, you can synthesize macrophages, you can synthesize that is develop eosinophil, basophil, and neutrophil. And this marrow adipocytes also showed here. Adipocytes are responsible for. How do you do this transplantation, bone marrow derived multipotent stem transplantation to treat hematological disease? What do you mean by hematological disease? Look, some people are affected by leukemia, leukocytes, the cancer full of blood cells, septicemia, otherwise called as your blood is damaged by cancer, that is called leukemia. Thalassemia, that is otherwise a part of genetic disorder. So what you have to do, you have to take the corresponding blood cell. So from donor, you have to collect the stem cells from the patient bone marrow or blood, any hematopoietic blood, that is umbilical, blood or peripheral blood, PBMC. From blood, because PBMC will produce monocytes, and stem cell of the bone marrow also you will be getting from the donor, and you have to process, as earlier I told you. How will you process? You have to process in a petri dish, plate, a plastic, where you have to give the corresponding mouse nutrient feeder layer, and feeder will provide like extracellular matrix and food, and culture media you have to provide, and you have to keep it for several months. And these are all processing method. I don't want to elaborate it in detail. And you have to cryopreserve for several months, and blood or bone marrow is frozen in cryopreserved in this case. And you have to give the chemotherapy or radiotherapy for the patient. After deriving the cells from the donor, you have kept this bone marrow or blood cells, stem cells in cryopreserved cells. Cryopreservation is done in a freezer, and before giving this cryopreserved cells into the patient should be administered with a high dose of chemotherapeutic agent and radiation therapy. Why? Because here allogenic problem there. What do you mean by allogenic problem? I will discuss later on the next slide. I need anyhow at least more than 1 hour to complete the topic. It's very interesting also to, if you are very much interested in doing stem cell research. So yeah, allogenic, I will give you what do you mean by allogenic stem cell. So first you want to know allogenic stem cell. What is this? Allogenic is nothing but sources of stem cell obtained from another donor, therefore completely unrelated donor or familial allogenic. Family member they can donate, or your friend unrelated donor also they can donate. These are all the stem cell they are produced from or donated from the people. These people are the stem cells are called as allogenic stem cell. And this situation, sometime infection will be, a transplantation process will be there. So they are extracted from the donor body or cord blood. So that's what here I want to say. Allogenic transplantation process, in order to avoid the rejection process, the patient is given high amount of chemotherapeutic agent or radiation therapeutic agent, and therefore that condition alone, this patient have the ability to receive the cryopreserved bone marrow stem cell or blood cells, and now the rejection is not possible. So what you have to do, you have to collect this cryopreserved cell into thaw. It should be thawed, and after thawing, the stem cells are reinfused into the patient, because the patient is already given chemotherapeutic dose of 5 dose of chemotherapy or radiation therapy in order to avoid the allogenic transplantation rejection process. Then, as I told you, explain what you mean by allogenic. From same another donor, your friend or family member donate your stem cell, then they are called as allogenic. In order to avoid, you have to give chemotherapy agent or radiation therapy agent. So this is nothing but your allogenic transplantation process. You have to collect from the donor and bone marrow or blood cell, and you have to do the processing, as I told you already. I don't want to repeat again.
and you have to do it for freezing, and the freezing is done by cryopreservation method. After freezing in cryopreservation method, the people are given, the patient are given the chemotherapy or radiation therapy in order to avoid the rejection process, and afterwards that stem cells are infused into the corresponding patient. Then the patient will accept the infused cell, that is otherwise called as allogenic stem cell from the donor. What do you mean by autologous cells? Auto means your patient own body. Say for example, this patient is here and need not depend on the donor cell for collection of the stem cell. So the patient from where you can get peripheral mononuclear cells, PBMs blood cell, or your bone marrow, and from that bone marrow you have to no, here there is no necessity of giving chemotherapy. If you take autologous stem cell, no need to give chemotherapy or radiation therapy, because rejection is limited or minimum or no nil rejection. So that only you have to cryopreserve for in order to mature the corresponding stem cell. After cryopreservation, you have to reinfuse into the patient. So they are called as autologous stem cell. In such case, no need to provide chemotherapy or radiation therapy. So this is very easy method. Unlike bone marrow harvest, it can take place outside the operating room, and patient does not have to be under general anesthesia. So there's all these things are carried out in the allogenic transplantation mechanism, whereas in autologous transplant, physician usually collects stem cell from the peripheral blood rather than bone marrow. And what do you mean with xenosis? Species is different. Say pig. From pig also you can take corresponding stem cell. Okay, so you might have heard about the pancreas cell that has been collected from the pig to synthesize the type 1 diabetic patient. The problem is type 1 diabetic patient lacking secretion of insulin comparing to type 2 patient. So the type 1 patient pancreas doesn't have the ability to synthesize more amount of insulin. In such case, transplantation takes place. So transplantation, the pig is a rich source of pancreas, which has the ability to transplant from pig to the patient, and it will synthesize automatically your insulin. In such case, you have to depend on other species. Therefore, it is called a xenogenic stem cell. So porcine, fetal, ventral, mesencephalic xenotransplants for this type of pancreatic or any type of brain disorder, mainly Alzheimer or Parkinson disease, you can also use this. But the problem is immunosuppression and rejection are the problem in the case of the xenogenic, because even in allogenic you are providing chemotherapy, except autologous, both allogenic and xenogenic stem cells are very risky transplantation method. In order to avoid, you have to be careful of immunosuppression or immunorejection process while it is transplanted. And similar to our collection of stem cell from umbilical cord, or otherwise, not umbilical cord, embryo, as I told, in pluripotent stem cell, you can also collect stem cell from bone marrow. This is a process, as I told you already. You have to open the anterior crest of the bone, sternum at the particular site, and you have to do some skin puncture. And you have to do it skin puncture where the patient need patient or the donor, whoever going to donate this corresponding what is called stem cell. You have to do the puncture in the iliocrest region of the bone marrow, and you will be getting bone puncture, so that you will be getting 10 to 15 milligram per kilogram. Our weight is 70 kilogram. So per kilogram can get 10 to 15 milligram, and calculate for under 70 kg of the patient and the recipient or donor, wherever you are getting this particular what is called bone marrow derived stem cell. Whichever less, you can collect this 10 to 15 milligram per kg, and it is collected in a parenized syringe. Then it should be filtered through the 0.3 to 0.2 millimeter screen in order to remove the fat or bony sphicolic, it's otherwise called contaminants, because it has, otherwise we will be doing centrifugation. In order to avoid the damage to the cells, we are not doing any centrifugation. So here, what you are doing, you have to screen whether you have any fatty tissue or bony spiculas there. Then further, based on the clinical situation, you can remove the RBCs to prevent hemolysis, because in transplantation processes, I told you allologous and xenologous, you'll be getting hemolysis process. So you have to remove the RBC. Mainly in the ABO blood group, this transfusion hemolysis is possible and mismatch is possible, and therefore you have to be very careful while adding the RBC while adding this particular ABO group of patient, and to prevent hemolysis. And immunocompetent donor T cells also very important. Because immunogenicity is very important in our system, because whenever you are getting some outer cell, the system will consider that particular cell as a antigen. So the particular cell should be competent to the host cell. So competent cells only you can take. So removal of immunocompetent donor T cell prevent this type of what is called graft rejection, graft versus what is called GVHT, I told you, okay. Graft versus host rejection. Graft versus host, otherwise it will cause the rejection process. Therefore, it is called as GVHT. And tumor cells also, sometime I told you, mutation is possible. So in order to avoid the mutation, you have to be very careful with the stem cell which has been taken from the adult donor cell, in order to avoid the contamination in the case of the autologous transplantation. And immediately you have to transfute into the corresponding patient after harvesting. And if you do a delay up to 24 hours, it will cause some consequences, adverse consequences. So that in order to avoid some adverse consequences, immediately you have to harvest the bone marrow from the donor. Okay, so this is collection of stem cell from peripheral, that is peripheral monolayer cells. And hematopoietic stem cell, this peripheral monocell blood is very important source for the synthesis of B lymphocyte, monocyte, T cells, and red blood cells, as well as other macrophages, dendritic cells. These peripheral blood cells are important source. And you have to treat it with the hematopoietic growth factor, because you need a growth factor for the growth of the corresponding stem cell. So you need nutrient. So hematopoietic growth factor are also added with this stem cell, and a combination of chemotherapy in order to avoid the rejection process. And otherwise you'll be getting like malignant tumor, that as I told you earlier, the carcinoma will be there. So in order to avoid the malignancy, cyclophosphamide based chemotherapy are advisable in the case of combination therapy of this hematopoietic stem cell along with this chemotherapy. Otherwise GCSF are used. And when you treat this patient, you are collecting the stem cell from the donor, and 4 to 6 days you have to keep this stem cell with the growth factor, and you will be collecting the stem cell within 2 to 4 hour session. So in the case of autologous transplantation process, more than 2.5 and to 106 CD34 cells per kg leads to sustained engraftment. So in order to avoid this, what you have to do, the transplantation should be set properly in autologous transplantation process when you collect the stem cell from autologous patient. And in the case of PBMC, it's very easy comparing to the bone marrow collection. And peripheral mononuclear stem cells are easier to collect than bone marrow stem cell. And in the case of bone marrow, you have to collect the stem cell within bone, whereas peripheral mononuclear cell, you can collect it from monocytes. It's very easy. So it's very invasive treatment option than bone marrow stem cell. So this is nothing but your collection of stem cell from BBMC, and peripheral blood cells can be obtained from the donor. You are collecting the corresponding blood. So lab scale level step-by-step process of stem cell culture, what you have to do? You have to, you are doing in culture plate, you have to choose the culture plate, and you have to add the corresponding nutrient and factors, and cell culturing is done. Cell is cultured here, the stem cell culture, that is wherever you have taken bone marrow or peripheral mononuclear cells, or any type of dental teeth, scalp, or wherever skin, or wherever source you have taken the corresponding stem cell, you have to culture. And this is nutrient plate, and you have to add the RPM. This is feeder cell. You'll be adding the feeder cell, and this is RPM media with nutrients, and keep it in the incubator. Why? Because we are also oxygenated with oxygen and the elimination of CO2. Therefore, in such case, CO2 gas is produced in the CO2 incubator. Therefore, this culture plate should be kept in the CO2 incubator. So what are the differences between embryonic versus adult stem cell? So the differences between the adult and embryonic stem cells are, here embryonic stem cells are known source, that is mainly ectoderm or endoderm or mesoderm, that is egg, that is other embryo. They are large numbers that can be harvested from embryo, and it will cause immune rejection, but rejection of embryonic stem cell by recipient has not been shown yet. But in the case of multipotent or pluripotent stem cell, the differentiation into cell types is limited. Here it can be differentiated to any type. Any type is nothing but pluripotent. And in the case of multipotent, limited. Sound. And unknown source, this is unknown source, you, because you don't know from where your adult stem cell, and limited numbers of stem cell will be produced. It is very difficult to isolate, and the immune rejection is very less because the patient own cells can be used. Okay, these are the different adult stem cells. And the next source is umbilical cord. As I told you, this is primitive stem cell compared to adult stem cells, and therefore these are able to expand rapidly, and it can be stored properly because it has been taken from child, and it will be matched with that child, because nowadays the couples, those who are newly married, they are during the stage of birth of the child itself, the educated new couples, they are registering for the cord blood cell storage. So the cord blood cell will have excess amount of stem cell. So that is the advantage of the educated couples. Nowadays they are registering their baby's cord blood cell into a bank, because in future if the baby gets some problem in their body because of genetic disorder or any type of accidental because of accidental disease or accidental cases, the organ will be damaged. In such case, if the parents go and find out the store the child stem cell from the bank, they can easily convert this stored cord blood cell from the
collect the cord blood cell from the bank, and the technician and the physician can transplant this stored blood cells into the convert into a particular organ where the damage is taken, and can be what is called transplanted into the child, the grown boy, otherwise the adult, because already the adult stem cell was stored in the child birth itself. So it can be implanted easily without any graft versus host disease. That's what I told, GVHD. Graft versus host disease is not possible if suppose if you stored your child umbilical cord or amniotic fluid of the child or the mother in the bank. So cord blood stem cell close match for sibling also or family members in case of need with low risk of graft versus host disease. So this is nothing but cord blood derived multipotent stem cell banking treating hematological disease. As I told you already, this is a master cell. You have the umbilical cord, taken from umbilical cord and
Prabhu sir, there 10 minutes only there. Time. So we
okay.
set up to 6:05. So
Okay,
complete
okay,
within
okay,
5
okay.
minutes. Okay, sir. Okay.
Okay, and here you have the umbilical cord, and this umbilical cord is containing rich amount of the what is called stem cell, and this is otherwise called a master cell, and this can be used for regeneration of any type of cells like heart to treat the disease or dementia. So you have to decide cells, family history of the, if suppose the parents, couples, family has a malignant or benign inherited disorder, they can store their body, the baby blood cord cells for future use, because the chances are there for getting the baby get a malignant disease or benign inherited disorder. If suppose if the baby gets in such case, if in the during the adult stage, if he or she gets this type of disease, you can use the baby cord blood stem cell using this collected baby cord blood stem cell to treat this type of disease. Similarly, health risk factors, if the present in the family, then you have to store the blood clot cells, then it can be easy for removal of this health risk factor. Similarly, it is affordable one, the blood clot cord blood stem cell, and it is also chance to collect them at initial level. So SS means if you have the chances of history of malignant tumor, health risk factor in your family history, then definitely the new couple, those who have come from that family, should store the umbilical cord of the baby or amniotic fluid cell into the bank. Then that will be useful for future health risk to remove such type of, to avoid such type of health risk for the baby or the adult cell. So this is nothing but collecting the cord blood cells. There is no pain in the case of the cord blood collection, and 5 minutes work only. There is no pain, harm or risk to mother or newborn, and easily it can be collect from the baby once it's born and cord is cut, and from there you will be getting the cord blood, either private or public, where it is processed stem cell is preserved in liquid nitrogen. Liquid nitrogen is the freezer where you can collect the store the cord blood storage, and this is cord blood storage liquid nitrogen. And the second method I do, since the time is very less and the administrator or organizer, they informed me to complete the session before 6, because already 6. I want to quickly go through the slides. I don't want to focus individually. So this is second method of collecting stem cell. You can also collect this from stem cell from what is called placenta. It's a tissue. The tissue also, umbilical cord tissue also, you can take cord blood, stem cell. And another disadvantage is after delivery, it's delivery care of the mother and infant is very important, and you have to cord blood collected should be stored. A single placenta is called cord blood unit, ranging from 60 to 120 ml only you can take. And this is umbilical cord application. Adult stem cell infant origin, greater compactability is less expensive. So these are all the advantages if you collect stem cell. And plasticity, that is, it can be converted to any type of cell. That's what plasticity.
plasticity, it other type of like liver cells, neural cells, any muscle cell or any type of what is called this okay, homing is also very easy. The site of tissue damage, engraftment is also easy. There is no rejection process. So problem in the case of umbilical cord. And used to pluripotent cell, this is recently discovered in 2007. It can be grown indefinitely. That is what it is called inducible. So you have to induce by factor. So the same type of adult stem cell you have to induce the same pluripotent. It can be converted into specialized cell, but you have to induce. It's like a genetic engineering method. It is induced type of method. The factors are you are artificially inducing this pluripotent. Therefore, it is called induced pluripotent. The advantage is that can create a directly stem cell from pair. This is nothing but your induced pluripotent cell. So from multipotent stem cell, you can convert this induced pluripotent stem cell. And you have to add some signals or biochemical factors along with stem cell to develop into matured cells. And application tissue repair. So now this is nothing but the last part of the slides. Tissue repair, the production of nerve, heart, muscle, organ, skin, cancer disease, autoimmune disease you can cure, diabetes and rheumatoid arthritis, drug testing, and it can also be used for therapeutic purpose. Desired drug you can choose to deliver the drug. You can choose stem cell, and it is also used for Parkinson disease type of this one, and limb organs can be synthesized. You can also help the scientists to learn about human. If you want to test the drug efficacy, you need some cell. For that, you can use stem cell, and you can find out the efficacy of the drug in the stem cell. And millions of scientists, they are using some potential drugs and medicine without the use of animal. You need not depend upon the in vivo or animal testing or human testers, clinical or preclinical study. Instead of using clinical or preclinical study, you can use this newly developed stem cells for the testing of potential drugs, and is also used for no need of depending upon the human embryo. So in order to avoid the consequences of birth defects, pregnancy loss, and infertility, you can use this umbilical cord type of this one, and mainly cell-based therapy, that is Parkinson, Alzheimer's, spinal cord injury, stroke, severe burns, heart disease, diabetes, osteoarthritis. And this is nothing but simply you are taking bone marrow from patient, and stem cells are harvested, and you have to treat with agents that destroy leukemia cells without harming the bone marrow or stem cell. And the bone marrow of the patient will be remain as such. After the treatment of the regenerative stem cell, it is injected into a patient, and you will be getting the genetically modified stem cell. So even patient-specific therapy also possible. So individual patient, that is precision medicine. The precision medicine also possible. Biopsy you are taking and transfer the nuclear, and you reprogram the nuclear of that particular biopsy, and culture the blastocyst stage. And now you are transferring the corresponding nuclear, reprogrammed nuclear into the pluripotent stem cell, that is embryonic stem cell, and convert it into blood, nerve, heart, and muscle, and transplant it to the specific patient. And similarly for ocular disease, that is high disease, you can use this. And brain disorder, Alzheimer's disease, you can use this. And mainly this Alzheimer's disease, since I don't have time, the people are urging me. I don't want to explain in detail. If I have detailed enough time, I could have explained you properly in detail process. And I don't want to explain it in detail now. And these are the possible use of stem cell technology: replaceable tissues, organ repair of defective cell type, delivery of genetic therapies, and chemotherapeutic agent. And disadvantages, you have to use this destruction as ethics. Blastocyst, if you are using embryonic stem cell, you are using blastocyst, that is 14 to 18 days baby. That is also unethical. So blastocyst is a human life now, therefore it is immortal. So this is unacceptable one. It is one of the disadvantage. And long effect cause interference with any other antigen. So this should be materialized. And the carcinogenesis, as I told you already, the cancer mutation process also possible. So that should be avoided. And immunorejection like allologous and xenologous. And obstacle, how to find right type of stem cell, how to put the stem cell in the right place, will the stem cell perform the desired function in the body, which can be differentiate protocol for many cell type have not been developed so far. All these obstacles are in there still in the stem cell research. ACR is nothing but stem cell research. All this to be we have to resolve. Then only we can go ahead with the complete stem cell therapy process. The promise of the future promise of the stem cell research, you can identify the drug target for the potential therapeutics and toxicity also you can find out from the cultured pluripotent stem cell. And differentiation, you can identify understanding the prevention and treatment of birth defect. You can produce different type of tissue for leukemia patient, bone marrow, and chemotherapy patient. You can also produce a nerve cell for Parkinson, Alzheimer. You can also produce heart muscle cell. You can also produce pancreatic islet cell for diabetes. And these are all the, I don't want to again say the ethical issues. Majority of the adult stem cell can be found in the human body, and the best source of the stem cells are fetuses or embryos, some people are telling, and some people are religious concern, and their problematic issues are fetal stem cell can be cultured from cells isolated from the aborted fetus, and they are telling like that, and the embryos are isolated from 5 to 7 day old blastocyst. So this is also one of the ethical issue nowadays, and both of the things should be sacrificed. And the blastocyst used stem cell research is microscopically small and has no nervous system. That is also one of the problem. And whether this particular ethically produced person or right to live comparing to other normal person. And does science have a view on this various religions say about different aspects, and religious views are different, and democratically make human public policy also there. And harvesting embryonic stem cell destroys the blastocyst, and this is they are telling murder of the embryo, and that is also one of the problem. And research requires human cells, embryonic stem cell, that is also very problem. Now if suppose if it becomes commercial, then it will be marketed, so it will be marketed as commercial human stem cell. This will devalue human life. This is also the problem. So this is cloning. You can take skin cell and unfertilized human cell, egg cell, and the nucleus, skin cell. In order to avoid this, you can clone. You can clone this skin cell and convert it into the remove this nucleus and take this nucleus from the skin and add into the egg cell, and the egg divides and use this blastopia, then convert add into the recipient. So you can avoid this type of problem, ethical issue in the case of therapeutic cloning. So it's a best method. Cloning human tissue has never been done, but one way it might be performed. So because you are not using the here the nucleus of the egg cell, only you are using the outer layer of the egg cell. Only you are taking the nucleus of the cell, and you are providing all type of environment for the generation of the stem cell to transfer into the patient. So take home message is, there are certain modern perspectives in which stem cell can be produced in an effective manner, and this particular science stem cell research also faces some ethical issues, faces ethical problem. And even though certain type of ethical problems are there, in order to maintain its modern perspective research process or advantageous process, research on stem cell is still progressing in spite of several restrictions, because the one is very important rather than the second one, because we need effective treatment for any type of diseases using stem cell. Therefore, the first one is taken care by not taking this particular point as a considered one. So take home message is, first one is important. Research on stem cell is progressing in spite of several restrictions of such type of ethical issues. And thank you all. I can be contacted by you through my official email ID, sprabhu@svcac.in. and I can be contacted through my my own email ID or YouTube Prabhu SSB to the email.com, and Facebook and Twitter, LinkedIn Prabhu Sumaran, and Twitter at Prabhu SSB 2008, and Instagram Dr. S Prabhu, and WhatsApp this 9840182. Thank you all. Thanks for your patient listening. And if you have any question, I can answer for your specific question.
Thank you, sir. Thank you for your wonderful session. That was very informative.
You are from where?
Sir, this is me, Bharati, PSR Engineering College.
Okay, okay.
Final year
Organizer, okay.
student, sir. Yeah.
If there is any question, please ask.
Yes, sir, sure. There are a few questions from the participants. The first question is from
Participants are
Jayamani
sent there
Kandan.
because one
Yeah,
minute.
sir, sure. They are.
The time is 6:50.
They are, sir.
Okay,
They are.
okay. Tell me.
The first question is from Jayamani Kandan Venkatachalam. What are
He
the
is from?
major factors?
He is from? Please introduce him. Where he is from? Where?
Sorry, sir, that I couldn't get it.
Okay, okay, okay. Then.
What are the major factors responsible for production of stem cells from adult cells?
Factors we have to consider immunogenicity and rejection process, and what is called donor is autologous donor or allologous donor. That also we have to search in order to get the donor in order to get the bone marrow stem cells. So these factors should be considered, as well as immunogenicity is very important, and the corresponding what is called efficiency of the stem cell from the bone marrow lineages. We have to find out if suppose the lineages of the bone marrows are having rich amount of the stem cell, then you can consider that particular bone marrow lineages for the collection of the bone marrow derived stem cells.
Thank you, sir. The next question is from Vishnupriya Seduraman. Despite seven
No, I am not getting your question. Some. Hello.
Sir, now I'm audible? Audible, sir?
Yeah.
Despite several types of stem cells discovered from germ cells, which promising form of stem cells will transform the future of regenerative medicine, as it raises many ethical, political, and public issues?
Okay. So that's what I told you. Ethically, embryonic stem cells are many religious, political, and people are religious people are politicizing that collection of stem cell from embryo. But when you consider medical field, the embryonic stem cells are good because it can be converted into any type of stem cell. It is otherwise called as pluripotent stem cell. And if any problem in embryonic stem cell, you can use in vitro fertilization. Using that in vitro fertilization, you can choose embryonic stem cell as a source for generation of majority of organs or tissues. So future, if suppose the people are educated, religious people are educated, or otherwise the political people are educated well, then the best choice is embryonic stem cell rather than adult stem cell, because sometimes the infection is possible, and in the case of the embryonic stem cell, otherwise the embryonic stem cell also takes many time for it is a specific stem cell. It can be modified into specialized stem cell. It cannot be differentiated into any type of cell. So otherwise, if there is no problem from the political people or religious people, adult stem cell as well as umbilical cord stem cells are very future stem cell usage for the generation of any type of organ in the case of regenerative medicine.
Sir, sorry, sir, we are facing some inconvenience as here it's raining heavily. We will post all the questions to your mail ID, sir.
Yeah.
No, yeah, for
Thank
me,
you,
no
sir. Thank you.
problem. No problem for me. If the audience are okay, no problem. I can answer. It depends. It depends on audience.
Okay, sir.
Mm.
Thank you, sir. Thank you for your
Thank you.
wonderful
Thank
session.
you so much.
Thank you for spending your valuable time with us, sir.
Thank you, ma. Thanks for the participant. Thanks for the organizers. Thank you so much.
Sir, thank you, sir. Thank you so much.
Thank you. Rajeshwari
It's a
madam,
wonderful session, sir. Yes, sir. Yes, I
Balamurugan
am here.
Karthi sir,
Hmm.
and Grace sir. Everyone, I am thanking.
Okay, sir. Thank you. Thank you
Thank you,
so
sir.
much, sir.
Thank you,
Hmm.
sir.
Thank you. Thank you so much.
Thank you, sir.
Okay, sir. Okay, sir.
Okay. Feedback can you send to me?
Thank you, sir.
Yes, sir. Surely, sir. We
Okay.
will send to you, sir.
Okay. Thank you.
Surely, surely. Okay, sir. Okay, sir.
Sorry, I took a lot of time.
Sir, then no, that's not a problem. What we faced means in GMIT first we set 2.5 hours up to 6 o'clock. That's the
Yeah.
problem.
Yeah.
After 6 o'clock, every place is raining, so we cannot
Yeah.
able to do anything.
Yeah.
Here, heavy thunder, storm, and also that's why
Yeah.
the
I
problem.
understand.
Otherwise, we can continue.
Yeah.
No
Yeah.
problem, sir.
So, okay.
Thanks
No.
for your acceptance, sir. You
Okay.
gave wonderful speech also.
Thank you. Thank you so
Because
much.
a very vast area, so you
Yeah.
covered all the areas, sir.
Because
Most probably, I think.
the participants are mixer of basic people.
Yes, sir.
And
Yes, sir. Yes. Everything is there. No, so that's why. Okay, sir. No problem. We will call you later, and then we will send the feedback also.
Thank you.
Okay,
Thank
sir.
you so.
That's not a problem, sir. Thank you so much for your acceptance and speech.
Thank you.
Thank you, sir. Okay, sir. Okay, sir.
So, thank you, sir.